Welcome to the CaeNDR Natural Diversity Resource, or CaeNDR. CaeNDR is an unparalleled resource for comparative genetics and genomics across free-living nematodes in the CaeNDR genus. In recent years, a large number of new CaeNDR species have been discovered. Across the genus, we now have 3 different species that reproduce primarily by self-fertilization: C. elegans, C. briggsii, and C. tropicalis. C. elegans has been extensively studied for more than 50 years, but the satellite model species of C. briggsii and C. tropicalis can be used for comparative studies. At CaeNDR, our goal is to enable studies of natural diversity for the community. Previously, CaeNDR focused primarily on the C. elegans species. During that time, numerous research groups have moved beyond the laboratory-adapted N2 strain and incorporated wild C. elegans strains, leading to many discoveries about human biology and evolution, including over 100 publications. These discoveries can only be amplified if we increase resources to include 3 different species of CaeNDR. Over the past 10 years, many researchers and citizen scientists have gone out into nature to look for new CaeNDR species and new strains in already characterized species. These efforts have paid off with over 1,500 strains of C. elegans and C. briggsii and over 700 strains of C. tropicalis identified so far. This collection of wild strains harbor millions of variants and can be used as a reservoir of natural diversity for all 3 species. It's time to move beyond C. elegans and the N2 strain. All of these strains have been cryopreserved in CaeNDR and are available to the community so that anyone can do their own studies of natural variation. We've built a series of tools to browse where worms are found in nature, environmental conditions at the time of isolation, and pictures of the natural substrate. You can collect your own wild strains and then submit them to CaeNDR to increase the natural diversity for the community. We have sequenced the whole genomes of all of these strains and identified variants to understand genotypic differences across these populations. For C. briggsii and C. tropicalis, we created new reference genomes and gene models so that we can more easily identify genetic differences across the wild populations and predict their effects on genes. All of these genomic data and other data relating to natural differences across the 3 species are available to download and browse. In collaboration with Northwestern IT, Media and Technology Innovation, we have created web portals to empower studies of natural variation for all 3 species. You can query your favorite gene to find out how natural variation impacts its function. All the wild strains are available to order and study in your favorite traits of interest. With a small collection of strains, researchers can collect replicate phenotype data for any trait, and then calculate how much of the trait variance is genetic. This so-called heritability can be studied using the heritability tool on CaeNDR. If genetics contribute strongly to a trait of interest, then users might want to measure phenotypes for more wild strains and perform a genome-wide association study, or GWAS. We built a GWAS mapping framework optimized for CaeNDR species that reproduce primarily by self-fertilization, called Nemascan. And this package is implemented in a web portal on CaeNDR. Users can enter their own trait data and perform a GWAS. A report of the mapping experiment will be generated showing quantitative trait loci, regions of interest for fine mapping, and suggesting candidate genes for future study. Once a genomic interval is identified using GWAS, users will want to narrow this interval to identify the specific gene that underlies natural differences in this population. This narrowing requires genetic markers that can be easily studied in the laboratory. So we built a tool to compare any 2 wild strain genomes and identify insertion, deletion, or indel variants that can be genotyped using PCR and gel electrophoresis. Indels are suggested along with primers to amplify the regions surrounding these indels. After PCR, amplicons of different sizes tell you what genome, either strain 1 or strain 2, is present at the location of the indel marker. As genomic resources for wild CaeNDR strains increase, CaeNDR will continue to enable studies of natural diversity. The power of nematode model species makes discoveries about human biology and processes of evolution possible. Please check out CaeNDR and let us know if you have any questions. Thank you.